Hey everyone, welcome back to CellsToSell. My name is Heem, and that's Nick, and we're back at it for another Trivia Tuesday. We hope you guys enjoyed our previous couple with the entrepreneurship and the environmental science one. This week we're going back to our science, the more biology science, and we're going to do a basic biochemistry one. Nick's idea.
This should be fun.
It's biochemistry is like so specific.
I want to watch Heem suffer.
Oh god.
That's why you're going first.
Oh. Okay. You know what? First is not good because Nick will end on the hard questions. I believe it. All right. Question 1. If you wanted to get a lot of quick energy before a soccer match, which biomacromolecules would your body need to break down? Proteins, carbs, lipids, or nucleic acids? I'm going to say carbs, right? Because the day before a big match or something, you eat pasta. So carbs. Correct. 1 for 1. All right. This is not bad, Nick. All right. 2. Complete the analogy. Lock is to key as enzyme is to substrate. Yes. The key goes in the lock. The enzyme substrate goes in the enzyme.
Technically, it's a modified lock and key concept when it comes to enzymes because the substrate changes and the enzyme changes in order to come in contact.
Me.
Just saying.
Master key.
Maybe.
All right. Question number 3. Unlike saturated fatty acids, unsaturated fatty acids contain what kind of bond between carbon atoms? Double bond. It's a double bond. Because that's what causes, if it's a saturated, that means it's a complete straight line of carbons and has single bonds between them and fully completed hydrogen orbitals. And then unsaturated means one of the carbons is not saturated with the amount of hydrogens it has. It has only one hydrogen link, so it causes the kink and the double bond. So yeah. Question number 4. Which are examples of carbohydrates? Starch, glucose, fructose, cellulose, glycogen, protease, cholesterol, wax, carcinogen, trypsin, lipase, testosterone. It's the first one. Starch, glucose, fructose, because carbs are sugars. Correct. Come on, Nick. Come on, Nick.
You were the one afraid to take this. I didn't think it'd be that bad.
All right. Question number 5. Which would not be a correct match? Keratin, amino acid, phospholipid, fatty acid, DNA, nucleotide, cellulase, monosaccharide. Well, cellulase would be a di or a polysaccharide, I think, because glucose, fructose, and something else are monosaccharides, I think. Phospholipid fatty acids, I would think so. But phospholipids, fatty acids have a NO, but I don't think they have a phosphorus. So I don't think it's fatty acids. DNA nucleotide, I think that might be the most... Oh, which would not be a correct match. Oh. Cellulase monosaccharide. Correct.
Cellulase is an enzyme, yeah.
Yeah. All right, question 6 is the last one for me. This picture represents what biomacromolecule? It is a nucleic acid. Because it has the carboxylic acid at the end. No, wait a second. This is not a nucleic acid, because a nucleic acid has got to have a nitrogen. It's not a lipid. Oh, wait, it does have a nitrogen. Ah! It's not a protein. I don't think it's a carb. I'm going to go with nucleic acid. No, it's a protein. What?
It's an NCC bond, man.
What, that's a protein to you?
Yeah.
Okay,
It's
whatever.
Alright, you went 1 for 6.
1 for 6. 5 for 6.
5 for 6, not 1 for 6. That's really bad. All right. My quiz, so hopefully I do well. All right, what is the function of lipids? They're used as cell receptors, enzymes, transport molecules in the human body? No. Used as main energy source for plants, stores hereditary information in bacteria, or used as insulation for marine Arctic animals? I'm going to go with D. Can be used as insulation. That is correct.
Nice.
On to question 8. In enzyme blank, the activation energy needed to achieve a chemical reaction. Does it stabilize, raise, lower, or eliminates? It lowers the activation. What is it? Delta G, I think is what it
Activation energy is?
yeah, I think so. I think. Lowers delta G. Okay. Question number 9. A biomacromolecule which consists of a polymer of fatty acids is a nucleotide, lipid, carbohydrate, or amino acid. I'm going to go with a lipid. That is correct.
Nice.
Yeah. The macromolecules of a nucleotide is nucleic acid, so RNA, DNA. Carbohydrate would be like starch, you know. A lot of carbohydrates would be like a starch, and then amino acids would just be a second degree protein or amino acid chain. Okay, question number 10. A feature of nucleic acids not found in lipids is oxygen, potassium, phosphorus, or hydrogen. Ooh. A feature of nucleic acids not found in lipids. So oxygen and hydrogen are obviously found in both. I think that it's gonna be potassium. I think so, because like the phospholipid, that entails a phosphorus. So potassium is my final answer. It is phosphorus.
Nice.
Maybe no. I answered potassium.
Potassium is K. Phosphorus is...
Right. A feature of nucleic acids not found in lipids.
Isn't potassium not found in lipids?
That's what I thought.
And what's the answer?
Phosphorus is the answer, and I answered potassium.
Phosphorus is found in... I mean, it is, though. Phosphorus is found in lipids.
That's what I'm saying.
That's so weird.
A feature of nucleic acids not found in lipids. Because nucleic acids had the phosphoric backbone.
Yeah.
Right?
Yes.
And lipids...
Backbone, yeah.
I guess it depends on what kind of lipid you're talking about. There's a bunch of different kinds of lipids, and not all of them are phospholipids. So, caught me on a technicality there. Good job, quiz.
But potassium, I don't know a single lipid with potassium.
Me neither. So, oh well. It's a fun quiz. I'm not getting a grade.
Fun.
So, question number 11. If you wanted to increase the rate of an enzyme catalyzed reaction, what would you do? So, increase the rate of enzyme reaction. Would you decrease the amount of substrate by 1/3, increase the pH by 4 levels on the pH scale, decrease the amount of enzyme by half, or increase the temperature by 10 degrees Celsius? So this is a weird question because how enzymes are affected by their environment changes by enzymes. So some enzymes work really well in low pHs while others work in really high pHs. And when you put them in opposite environments, they're actually going to shut off. Like they literally don't work. They have to be in the perfect temperature and pH for them to be their most efficient. And that's why sometimes when you get sick, you have different enzymes that are dormant when you're healthy. So when your body temperature is around like 99 degrees Fahrenheit. But when you get sick and your temperature rises to about 100, 102, you actually have different enzymes kick on the immune system faster. So this is kind of a weird question, but I'm going to go with, so if you wanted to increase the rate of an enzyme catalyzed reaction, I feel like you would have to decrease the amount of enzyme and leave the substrate at the same, because if you're too saturated, if you're too saturated by one of them's not going to move fast enough. So if you decrease the amount of enzymes, they're going to work faster and you're going to have a faster output is my thought process on it. And you increase the temperature by 10% Celsius, or 10 degrees Celsius. So
I would say increase the temperature.
But I feel like it depends though, because that 10 degrees Celsius could denature that enzyme. It really could. 10 degrees Celsius is a lot. That's like 40 degrees Fahrenheit or something stupid. So I feel like if you decrease the amount of enzymes and you had the same amount of substrate in there,
they don't move faster though. It's
I guess
their amount.
This is saying that increasing the temperature will make the molecules move faster, thus increasing the amount of collisions between substrate and enzyme, which also makes sense.
Yeah.
But 10 degrees Celsius is a lot. Is a lot.
It was a trick question.
That was a weird question. I get what they're saying. I totally
Yeah.
understand the question and why I got it wrong.
Yeah.
But oh well. This is my quiz.
Yep.
All right, question number 12. Which is odd here? Nucleic acid, monosaccharides, lipid, or protein? I'm going to go with protein.
Really?
Yeah.
I was going to say nucleic acid.
They're all building blocks. Monosaccharide builds starches or carbohydrates. Nucleic acid builds like DNA. Or I guess those would be
nucleic acid.
No, monosaccharides is the wrong one. Yeah, that's correct. I had to really think about that for a second.
Wait, why though?
Because nucleic acid is built of nucleotides, proteins are made of amino acids, and lipids are made of fatty acids. Yeah, and monosaccharides are just monosaccharides. That would be your glucose, maltose, and
the other ones are
galactose.
chains. This monosaccharides are like molecules.
Yeah. Glucose, fructose, and maltose, right? Those are the
Sucrose?
monosaccharides. No, 2 fructoses make a sucrose.
Okay.
Last question. This is giving me flashbacks to junior year. No, don't know why we took this. An appropriate enzyme to catalyze a reaction involving the imaginary substance camelcine would be camellose, camelline, camellase, or camellase.
Oh, I like it.
I'm going to go camellase.
To break it down?
The appropriate enzyme reaction involving the imaginary substance. It's just saying an appropriate enzyme to catalyze a reaction involving.
ACE.
So it would be ACE. Camellase. That's correct.
Nice.
All right, come back on in here. 77 out of 100. I think we got 3 questions wrong. So 2 on a technicality, 1 on a mystery. But I think that's okay. And again, this stuff is happening every single second in our body. And we've said this before, but when you think about regenerative medicine, we think about the big picture, all these cells interacting. But it's important to understand that the biochemistry of these cells is what makes them run. It's the engine. Mm-hmm. So I thought it was a fun quiz. It's good to challenge ourselves. We will eventually get a 100 on a quiz again.
That's funny.
Yeah, that's all I got for this one. You got anything else, Heem?
If you guys have any suggestions for quizzes, if you have any quizzes you've made, if you have any links to Quizlets that you've made for your classes, if there's any old exams you've had from your classes you want us to take and you want to send us PDFs, that would be so fun, but
that'd be awful. But we'll do it.
We'll do them.
So
that's all, though.
All right. Well, this has been another Trivia Tuesday, and this has been CellsToSell. I'm Nick, and that's Heem, and we'll see you guys in the next one.
See you guys.